Hello, I'm Kayla Lee. Through experiencing the pandemic, I've come to understand the importance of diagnostics. I'll present an innovative multi-diagnostic kit for emerging mosquito-borne diseases in Central and South America. Unpredictable weather patterns due to global warming cause the spread of mosquito-borne diseases such as dengue and malaria due to the change in habitats and behaviors of mosquitoes. The two most common types of malaria are Plasmodium falciparum and Plasmodium vivax. The initial symptoms are similar, but Plasmodium falciparum has a higher fatality rate after infection with severe symptoms. On the other hand, Plasmodium falciparum spreads more easily because it can develop in the vector in lower temperatures. In areas where Plasmodium vivax is already prevalent, potential spread of Plasmodium falciparum can increase the risk of mortality and cause difficulty in tracking the spread of Plasmodium falciparum. Due to the similarity in early symptoms between dengue and malaria, rapid and accurate differential diagnosis is essential for timely treatment and effective control of infectious disease spread. In the 2000s, dengue was prevalent near the equator, but dengue spread to high-latitude regions such as Latin America by 2023. Also by 2023, malaria dispersed, alerting that mosquito-borne diseases are spreading due to global warming. Since climate change cannot be resolved in short term, urgent preparedness is needed to address the spread of diseases caused by mosquito expansion. Conventional diagnostic technologies distinguishing between dengue fever and malaria require precise diagnostic techniques that rely on skilled operators and a stable power supply. Moreover, confirming the diagnostic results require a significant amount of time. Because such methods are difficult to implement in regions with limited healthcare systems, I aim to develop a diagnostic method that can be used even in these resource-limited areas to improve disease management. I invented a new diagnostic method for mosquito-borne diseases that have similar initial symptoms because prompt and accurate diagnosis helps begin the right treatment sooner, which can improve outcomes and prevent complications. Early detection also reduces the spread and saves resources. In order to be operated in low resource settings, the new diagnostic method should not require skilled technicians or equipment such as thermocycler, but visually indicate the pathogen-specific detection. Therefore, LAMP reaction may be an alternative because LAMP does not require high equipment requiring high power consumption, such as thermocycler, and allows results to be visually confirmed with the naked eye through the usage of colored dye. LAMP is a highly specific DNA amplification that uses multiple primers. Each primer is designed to recognize distinct regions of the target sequence. Conventional PCR requires expensive equipment and longer time, which is a challenge in Latin America. LAMP reaction can specifically diagnose infection within 60 minutes at a constant temperature, which simplifies the diagnostic process and overcomes the limitations of conventional PCR. Although primer set for LAMP reaction requires more than that for conventional PCR, if primers are optimally designed, it can work even in resource-poor environments with high specificity and sensitivity. The purpose of the study is to develop a diagnostic method to distinguish between dengue fever and two malarias simultaneously in a single tube through color change, for the purpose of analyzing and comparing the distributions of pathogens and mosquitoes to cope and prevent. Experiments were conducted to establish the optimal conditions for LAMP to enable multi-diagnosis. To this end, I designed the optimal primer sense to specifically amplify either dengue or two malaria genes, respectively. Because direct handling of the pathogen had an infectious risk, I needed an alternative that didn't handle the pathogens directly. So I decided to use the plasmid DNA of each pathogen for a template because DNA itself cannot cause infection. The usage of DNA in LAMP is not suitable and justified because it is an amplification method specifically targeting and amplifying DNA without the need of specific conversion. And then I designed a primer set for each pathogen's DNA. I verified DNA templates in each primer set to check that primers specifically amplify each DNA through conventional PCR and LAMP. Outer primers are used to amplify target DNA regions. First, and confirm whether each outer primer set specifically amplifies the target gene region using a conventional thermocycler. Figure 1 shows DNA size reference in the first lane. A band at each primer column indicates that primers successfully amplifies the fragment. No band appeared at non-corresponding or non-target lanes, showing that the amplification was specific. The results showed that each outer primer set specifically amplified the target DNA, indicating that each primer set was designed to be target-specific. This suggests that disease can be specifically diagnosed using LAMP primers. The color of magnesium ion bond HMB is purple, and the color turns sky blue when magnesium ions are separated during DNA amplification. Since BSD DNA polymerase consumes the ion for its activity, the color change of the LAMP reaction product reflects DNA amplification. Because the multi-diagnosed ticket is indicated by the color change, ensuring that color change correctly reflects amplification is important. In order to observe the effect of each component on HMB, the color of HMB was observed after each experimental component was added. Except for the reaction buffer containing magnesium ions, other components did not affect the color change of HMB, showing that the order of steps has no effect. Betaine is known for its ability to reduce the formation of secondary structures and stabilize DNA polymerase activity. Therefore, betaine is used for DNA amplification. I verified whether betaine affects H&B color reaction. DNA fragments were amplified even without H&B in addition of betaine, but betaine enhanced the blue color change. Therefore, betaine had no impact on the LAMP reaction, but led to a more stable and consistent blue color. It means that the addition of betaine can improve the accuracy of diagnosis through color change. Diagnostic speed depends on the duration of diagnostic procedures and the earliest point at which infection is detectable. To determine the diagnostic period and timing using LAMP, the optimal reaction time was determined. A color change began at 30 minutes, becoming the strongest and distinct by 45 minutes. Determining the color change with the naked eye can be subjective and cause error. To address this, an objective method was used. The color change from purple to blue was numerically analyzed with an application called color detector. Since the app provides numerical values for the intensity of primary colors, it allows for a more accurate and objective analysis of the color change. The hypothesis was that a smartphone application like Color Detector could convert visible color changes into objective digital data using the camera. This will help ensure the objectivity of the results. Therefore, Color Detector app was used to analyze the color change digitally during the experiment. As DNA was amplified, red intensity decreased, indicating a positive reaction. The minimum detectable concentration of the template was determined for identifying low-level or early-stage infections, especially when symptoms have not yet appeared. In the early stages, the pathogen has not multiplied extensively, resulting in a low pathogen concentration in the human body. Therefore, in order to diagnose infection early, it is important that low concentration samples can be amplified. The template was diluted in multiple times to create different concentrations of DNA template to perform LAMP. The color change was observed starting from 10 picogram for dengue, 1 picogram for Plasmodium falciparum and Plasmodium vivax, showing that the minimum concentration is 1 picogram. The minimum amount of sample amplified by conventional PCR is typically 1 to 10 nanograms. This indicates that physical time required for a diagnosis and the earliest detectable point using LAMP is not significantly different from that of conventional PCR-based diagnosis. Diagnosis using conventional PCR requires a stable electric supply of approximately 200-300 watts, but in rural or resource-limited regions, it is very difficult to ensure such a stable power supply. Compared to strict temperatures maintenance in harsh conditions, flexible temperature tolerance allows for a certain level of unstable power supply. Therefore, I examined the LAMP reaction under varying temperature conditions to verify the stability of LAMP reaction under temperature variations. Reaction occurs at temperatures between 45 degrees Celsius and 65 degrees Celsius, but not at 37 degrees Celsius with at least 100 picogram of template. This indicates that temperatures lower than 45 degrees are not suitable. However, diagnosis based on the LAMP reaction indicates that a certain level of variation within a range in reaction temperature is permissible. Low reaction specificity can cause problems such as amplification of non-target DNA regions, which may lead to false positive results, and the specificity of the results is determined by the primers. So I verified the specificity of the LAMP primer by conducting LAMP with a template of a different pathogen. The dengue and Plasmodium falciparum primer specifically amplify the DNA templates, while Plasmodium falciparum primer amplified both Plasmodium falciparum and Vivax templates. This means that the dengue diagnosis can be achieved with a single primer set, while distinguishing between different types of malaria required two different primer sets. To reduce the cost of the diagnostic method, LAMP reaction was performed with different concentrations of DNA polymerase. Positive tubes containing at least 0.25 microliter of DNA polymerase turned blue. LAMP reactions were successful even with 2 units, which is 1/4 of the recommended amount. This reduction can significantly lower the cost of the diagnostic kit and allows for accurate diagnosis with the minimum concentration of polymerase. Visual diagnosis by the naked eye can lead to inaccuracies due to subjective judgment. Therefore, an objective evaluation standard is essential, and I believe that AI can provide the solution. I collaborated with experts to develop a deep learning AI model, which was trained to interpret amplification results based on color changes. As AI was trained repeatedly, the number of epochs indicating failures decreased and success increased. This shows modeled strong predictive ability. Ultimately, only 2 negative samples were misclassified as positive in the confusion matrix, resulting in an over accuracy of 97%. This successfully identifies with the pathogen, patients with the pathogen and reduces the risk of false negatives. The multi-diagnostic kit will consist of 4 strips containing specific LAMP primers. When a specimen is dropped in the kit, it will migrate across the strips and interact with primers. After incubation at 55 to 65 degrees Celsius for 45 minutes, the pathogen's type will be indicated by a color change on the strips. This color change can then be accurately identified using the AI deep learning model developed in this study. Thank you for your attention.